Have you ever wondered how our bodies know how to create more cells? Just pause for a moment and think about it. Each day, our bodies are working tirelessly, producing millions of cells. But how does this incredible process occur? The secret lies in the intricate world of DNA and RNA. These two miraculous molecules serve as the blueprints for life, governing everything from the color of our eyes to our ability to digest certain foods. They're the conductors of a symphony of life that plays out within each of our cells, guiding the process of cell division and protein synthesis. Imagine a vast library filled with books. Each book contains a unique story, each sentence a specific instruction. That's what DNA and RNA are to our bodies, an extensive library of instructions that guide the creation and function of every cell. So, are you ready to delve deeper into this fascinating world? Today, we will explore the fascinating world of DNA and RNA. To understand DNA, picture a twisted ladder or a double helix. This helical structure is like the architectural framework of life itself, and it's as fascinating as it is complex. Now, let's break down the structure of this biological masterpiece. The sides of this twisted ladder are made up of sugar and phosphate molecules. These sugars and phosphates are like the scaffolds that give DNA its shape. Then, we have the rungs of the ladder, which are composed of two complementary chemicals or bases that pair together. There are 4 types of these bases in DNA: adenine, thymine, cytosine, and guanine. In the DNA double helix, adenine always pairs with thymine and cytosine always pairs with guanine. It's like they're dance partners, always twirling together in this beautiful ballet of life. These pairs are known as base pairs, and they are the heart of DNA's structure. But what's even more fascinating is how these base pairs work. Picture each base pair as a coded message. The order in which these pairs line up along the DNA strand spells out specific instructions for building an organism. It's like a recipe book, detailing what ingredients are needed and in what order to add them, to whip up something as simple as a bacterium or as complex as a human being. This is where we see the function of DNA in action. DNA stores these genetic recipes, passing them down through generations, ensuring that life continues in all its diverse forms. Whether it's the color of your eyes, the shape of your leaves if you're a plant, or the number of legs you have if you're an insect, it's all written in the DNA. So, DNA is like the blueprint of life, containing instructions for building every part of an organism. It's a wonder of nature, a marvel of science, and a testament to the intricacy and beauty of life itself. While DNA is the master blueprint, RNA is the builder who reads those blueprints. RNA, or ribonucleic acid, is a molecule similar to DNA. It's a crucial player in the world of genetics, serving as an intermediary between the DNA in your nucleus and the protein-making factories in your cells, the ribosomes. But before we delve into its role, let's first understand its structure. Unlike the double helix structure of DNA, RNA is generally single-stranded. It's composed of a sugar called ribose, phosphate groups, and 4 types of nitrogenous bases: adenine, guanine, cytosine, and uracil. Yes, you heard it right. Uracil replaces thymine in RNA. Now you might be wondering, why does RNA even exist if DNA already carries our genetic information? Well, consider DNA as the original copy of a precious book. You wouldn't want to risk damaging the original, would you? That's where RNA comes in. It's like a photocopy of the book, allowing the original to stay safe and intact. You see, RNA is the one that ventures out of the nucleus, carrying the genetic message from DNA. It's like a messenger, a courier, if you will, with a very important package: the instructions for making proteins. There are different types of RNA, each with a unique role. Messenger RNA, or mRNA, for instance, carries the genetic information from the DNA to the ribosomes. Transfer RNA, or tRNA, brings the correct amino acids to the ribosome to be assembled into proteins. And then there's ribosomal RNA, or rRNA, which is a part of the ribosome itself. Each type of RNA works together to ensure that the right proteins are made at the right time. They read the genetic code in DNA and translate it into a language that the ribosomes can understand, a process known as protein synthesis. So RNA, in a way, is the messenger that carries instructions from DNA for controlling the synthesis of proteins. How does DNA ensure that new cells have the same genetic information? This question brings us to the fascinating process of DNA replication, a vitally important mechanism in the life of a cell. DNA replication is a lot like a production line in a factory, where the original DNA strand is the blueprint, and the end product is a perfect copy of that blueprint. The process is meticulously precise, as even a single error could lead to significant problems like genetic disorders. First, let's talk about unwinding. The DNA molecule is a twisted ladder or a double helix, with the rungs made of pairs of chemicals called bases. To replicate, this ladder has to be untwisted and unzipped down the middle by enzymes. This creates two separate strands, each of which will serve as a template for creating a new strand. Next up is base pairing. Each strand of DNA is made up of a sequence of 4 different bases: adenine, thymine, guanine, and cytosine. During the replication process, free-floating bases in the cell are attracted to the exposed bases on the original strands, following the rule that adenine always pairs with thymine and guanine always pairs with cytosine. This ensures that the new strands will be identical to the original ones. Finally, we get to reassembly. The new base pairs are bonded together, and the 2 new DNA molecules wind themselves back up into the familiar double helix shape. Voila! We now have 2 identical DNA molecules, each ready to go into a new cell during cell division. So, in a nutshell, DNA replication is a finely tuned process of unwinding, base pairing, and reassembly. It is through this process that DNA ensures the faithful transmission of genetic information from one generation of cells to the next, preserving the continuity of life. Through replication, DNA can pass on its instructions to new cells. This is not just a biological fact, but also a testament to the precision and beauty of life at its most elemental level. How does RNA translate the DNA's instructions into proteins? This question ushers us into the fascinating world of protein synthesis. This process is a 2-step dance, comprising transcription and translation, choreographed by the RNA molecules. Let's start with transcription. In the nucleus, a segment of DNA unwinds, revealing the coded instructions for a particular protein. This is where messenger RNA or mRNA steps in. It copies the DNA's information, creating a mirror image of the gene sequence. This is very much like a spy copying secret codes. Once the mRNA has completed its mission, it leaves the nucleus, carrying the genetic message to the cytoplasm. That's the first part of the dance. Now, for the second part, translation. This is where the mRNA's message gets turned into a protein. But how does this happen? Enter transfer RNA or tRNA and ribosomal RNA or rRNA. The tRNA molecules are like interpreters. Each tRNA has an anticodon, a sequence that matches up with the codons on the mRNA. When a tRNA molecule finds its matching codon on the mRNA, it binds to it, bringing along an amino acid. Now picture the ribosome, made up of RNA and proteins, as a bustling factory. It moves along the mRNA, organizing the tRNA molecules and their amino acids into a chain. This chain, as it grows, folds into a complex 3-dimensional structure. And voila, we have a protein. So in essence, mRNA is the messenger, carrying the DNA's instructions to the cytoplasm. tRNA is the interpreter, reading the mRNA's message and bringing the correct amino acids. And RNA is the factory worker, assembling the amino acids into a protein. Each of these players in the RNA family has a distinct but interconnected role in protein synthesis. They work together to ensure that the DNA's instructions are accurately translated into proteins, the building blocks of life. RNA thus plays a crucial role in turning the genetic code into functional proteins. Now that we've explored the roles of DNA and RNA, let's recap. DNA, the blueprint of life, encodes the instructions for all the proteins your body needs. RNA, on the other hand, is the messenger that carries these instructions from the DNA to the rest of the cell. One key point to keep in mind is the differences between DNA and RNA. DNA is double-stranded while RNA is single-stranded. DNA uses the sugar deoxyribose while RNA uses ribose. And DNA contains the base thymine, whereas RNA contains uracil. For exam success, remember the steps of DNA replication and RNA's role in protein synthesis. DNA replication follows the pattern of unzipping, copying, and zipping. Protein synthesis involves transcription and translation processes. To make your study more efficient, try drawing diagrams to visualize these processes and use mnemonic devices to remember key terms. Remember, DNA and RNA are the language of life, and understanding them is key to understanding biology.